Good morning to everyone. My name is Evelyn Sánchez, and the title of my work is In-Depth Catalog of Regulatory Interactions for Trichoderma, atroviride, and Botrytis cinerea. Botrytis is the causative agent of grave mold disease and can affect more than 1,000 plant species, causing severe economic losses in crop production. On the other hand, Trichoderma, an endophytic fungus, is widely used as a biofertilizer and biocontroller against pathogens such as botrytis under little-known genetic regulation pathways. A gene regulatory network is a collection of relationships between molecular entities that interact to govern the gene expression, determining different cellular regulatory programs and cell fate. The gene regulatory network approach could be a good starting point to study this kind of interactions. Taking into account all the background information and also the catalog of transcription factors for Trichoderma and Botrytis presented at this conference, the motivation to develop this research is the models of the gene regulatory networks for both fungi have yet to be built, and without these gene regulatory network models, it is impossible to study the transcriptional regulatory events in different contexts, even more so at the genomic scale. Using protein sequences from fungal model organisms and experimentally regulatory information from their transcription factors, we obtain DNA binding site preference motifs as position weight matrices for Trichoderma and Botrytis. Taking promoter sequences from each Trichoderma and Botrytis gene and the PWMs obtained above, through FIMO we calculate all regulatory pairs of transcription factor and their target genes. Using the transcriptional data and a tree-based ensemble method algorithms such as Gene3, we filter the network, leaving the regulatory pairs supported by transcriptional information. With the gene regulatory network models constructed, two different RNA-seq datasets we used to construct the gene regulatory networks under constant light and the gene regulatory networks of fungal confrontation, validating the method and the gene regulatory network models. As a main result, we obtained the reference models of gene regulatory networks composed of 11,700 nodes for Botrytis and 11,697 nodes for Trichoderma. For Botrytis, we obtained 157,040 regulatory pairs, while for Trichoderma 178,197 regulatory pairs were obtained. Under continuous light treatments, both fungus show similar regulatory responses and activate orthologous genetic programs commanded by key orthologous TFs. The confrontation treatment caused a differential regulatory responses in both fungus. A hierarchical TF cascade controlling peptidases genes in Trichoderma was differentially expressed, while in Botrytis the TFs differentially expressed controls oxidative stress-related genes. The gene regulatory models obtained in this work will help both community of fungal researchers to generate novel hypotheses on different transcriptional regulatory events.